There is a sound your skull makes that you have never heard because you are the one making it. Somewhere behind your eyes right now, in the specific dark that only exists inside a head, something is going. It was going before you started reading this sentence. It will still be going after you stop. Nobody turned it on. There was never a moment where it was off and then on. No switch, no first spark. Just an unbroken hum that started before you had a self to notice it with, and has not paused since. Not once. Not even in the deepest, most dreamless parts of sleep. You have spent your entire life standing next to the loudest thing you own and hearing almost none of it. Here is the part that should probably bother you more than it does. Your brain does not wait for something to happen before it starts talking. It is never actually silent. Even in a sealed, lightless room, even with your eyes shut and your body still and nothing coming in from the outside world at all, the cells in your head keep firing, keep sending, keep having something, like a conversation with no one in particular. Scientists have a bland little term for this, spontaneous activity, which is exactly the kind of phrase that hides a genuinely strange fact behind a shrug. Your brain was never waiting for permission to begin, and somewhere in you right now, something almost said your name, not out loud, not in words. But there is a chance, not a large one, but a real one, that in the last few seconds a specific, ordinary word almost surfaced somewhere in the folds behind your forehead, and then didn't. This happens constantly and nobody tells you about it, because there's no good way to tell someone about a thought that didn't finish. If that has ever happened to you, a word arriving halfway, a thought you can feel the shape of but not the content of, gone before you could turn toward it, you already know something about your own mind that most explanations skip straight past. You just never had a name for it. We're going to spend some time in there, not to map it, not to label every room, just to sit with it long enough that some of what it's doing stops sounding like machinery and starts sounding like what it actually is, which is closer to weather than to wiring. Before any of that, though, there's something small to mention. This is Sleepy Science Channel, said the way you'd mention it if someone asked what you were listening to and you almost forgot the answer yourself. The fact that this is still here, still going, this many episodes in, is not something anyone takes for granted. Not the people who make it, and, it turns out, not the people who keep it running by supporting it, quietly in the background, the way you keep a porch light on for no one in particular. If you've ever found yourself wanting to keep something like this alive a little longer, there's a way to do that, and it's easy to miss, tucked wherever these things get tucked. Anyway, that's not why we're here. We're here because something in you is always speaking, and you have almost never listened to it directly. So start there. Start with the fact that underneath the version of you reading these words, the version with a name, a mood, a body temperature, an opinion about whether the room is too warm, there is a second population, billions strong, that has been having its own conversation this entire time, in a language that doesn't use any of the words you know. They don't look like much on their own. A neuron under a microscope resembles something a child might draw if you asked them to picture a tree that had given up trying to be tidy. A central body with wild branching extending outward in every direction, and one long, unlikely arm reaching further than any of them, sometimes shockingly further, a single fibre that can stretch In some cases, the length of your leg from a cell body sitting near your spine. There are roughly 86 billion of these in your skull. 86 billion is not a number anyone actually feels. It's closer to a number you nod at. That number has its own strange history, worth a brief detour. For decades, the figure most commonly repeated in textbooks and casual conversation was a much rounder one. 100 billion neurons, a number that turned out, when anyone traced it back far enough, to have no clear original source at all, more folklore than measurement, endlessly copied from one publication to the next, without ever being properly checked. It wasn't until 2009 that neuroscientist Susanna Herculano Hausel developed a method precise enough to actually settle the question, dissolving whole brains into a uniform liquid suspension of individual cell nuclei, a technique she and her colleagues have described, with disarming honesty, as brain soup, then counting a measured sample of that suspension under a microscope and scaling the result back up to the whole organ. The real number turned out to be noticeably lower than the old folklore figure, roughly 86 billion, not 100. And the method turned up something else, at least as strange as the correction itself. The overwhelming majority of those 86 billion neurons, something on the order of 80%, are not in the cortex at all, the wrinkled outer layer most people picture when they imagine a brain doing its thinking. They're packed into the cerebellum, a comparatively small, densely folded structure tucked underneath the back of the brain, traditionally associated mainly with balance and fine motor coordination, and, until fairly recently, mostly written off as cognitively unimportant. Whatever thinking actually is, in raw cell count terms, most of the hardware involved isn't where popular imagination has always assumed it would be. That reassessment of the cerebellum has kept gathering evidence in the years since. Patients with damage confined specifically to the cerebellum, once expected to show problems only with balance and coordinated movement, have also been documented showing measurable difficulties with certain aspects of language processing, emotional regulation, and even the kind of mental flexibility involved in shifting smoothly between tasks. Findings that would have seemed almost nonsensical under the older, narrower view of what that structure was for. Some researchers now describe the cerebellum less as a specialized movement module bolted onto the side of the real thinking apparatus, and more as a kind of general-purpose refinement circuit, one the rest of the brain borrows for whatever needs smoothing out, whether that's a hand reaching for a cup or a sentence being assembled in real time. It's a fairly recent, still developing correction to a picture of the brain that most non-specialists never got the memo about updating. What matters more than the count is what they're doing with the time, and what they're doing underneath everything is refusing to sit still. Even a neuron doing nothing at all, not part of any task, not processing any sensory input, not contributing to any thought you'd recognize, is not actually at rest. It is holding a charge. Its inside sits at roughly negative 70 millivolts compared to the fluid outside it, a tension maintained by tiny pumps in the cell membrane that spend enormous amounts of the body's energy, constantly, just to shove sodium ions out and haul potassium ions in against the grain, uphill, for no reward except staying ready. This is not a passive resting state. It costs the brain about 20% of your entire body's energy budget just to sit there, primed, doing what looks from the outside like nothing, and it doesn't stay quiet. Left alone, With no outside stimulation at all, neurons drift. Their internal charge rises and falls on its own, nudged by tiny leaks in the membrane, random openings of channels that let a few ions slip through where they shouldn't. The whole system trembling gently around its resting point, the way a full glass of water trembles on a table even when no one's touching it. Every so often, purely by chance, enough of that drift lines up in one direction, and the cell crosses its threshold and fires anyway. Not because anything happened, but because nothing happening is never actually nothing. Not at this scale. Researchers who study developing brains have found this kind of firing showing up before a fetus has ever opened its eyes, before there's been a single sound to hear or a single hand to feel. Spontaneous, self-generated bursts of activity rippling through the retina and the spinal cord and the earliest folds of the developing cortex, wiring the system before there's anything from the outside world to wire it to. You were talking to yourself before you had anything to say. Exactly when that talking to itself becomes something anyone would call experience, rather than simply unconscious biological activity, is a question neuroscience genuinely cannot answer yet, and it's worth saying so plainly rather than smoothing over the gap. Researchers studying fetal brain development have tracked the gradual formation of thalamocortical connections, the pathways linking the thalamus to the cortex, widely considered a minimum structural requirement for anything resembling conscious experience in the frameworks discussed later in this account, typically forming sometime around the middle of gestation, though the mere presence of the wiring says nothing certain about whether it's actually running anything experience-like yet, any more than a house having its electrical wiring installed tells you whether the lights happen to be on. This isn't a fact this account is equipped to resolve, and no honest one currently could. It's simply one more place where the confident, tidy language often used to describe the brain quietly runs out, and the actual state of the science is closer to a careful, ongoing argument than a settled answer. Some of the most convincing evidence for this constant background hum comes from people who lost a limb and can still feel it. Phantom hand, clenching, aching, present in every sensory way except the one where it's actually attached to a body. For a long time, this was treated mostly as a curiosity, a strange leftover glitch. It's better understood now as something closer to a direct demonstration. The region of the brain that used to represent that hand didn't go quiet just because the hand disappeared. It kept firing. It kept generating the sense of a hand being there, because generating a felt sense of the body was never something that required an actual signal arriving from outside in the first place. It only required the machinery that was already running on its own, the way it had been running the whole time, hand or no hand. The same basic principle shows up in tinnitus, a persistent phantom sound with no source anywhere in the world, generated entirely by activity inside the auditory system itself. A ringing that isn't arriving from anywhere because it was never going anywhere to begin with. Sit in a sealed, soundproofed room long enough, the kind engineers use to test how quiet can actually get. Walls built to absorb almost every reflection, so still that people report being able to hear their own heartbeat, their own blood moving, and most people, within 20 or 30 minutes, start hearing things that aren't there. A hum, a hiss, sometimes voices, sometimes music, faint and half-formed, the auditory cortex apparently unwilling to sit in true silence, and simply generating something, anything, rather than reporting back an honest absence. Nature, and apparently your own skull, abhors a vacuum. Given the choice between silence and static, it will pick static almost every time, and it will not ask you first. None of this is malfunction. This is the baseline. This is what a brain is doing before it does anything else. The low continuous hum underneath every specific thought you've ever had, present in the room the entire time you thought the room was empty. None of it either belongs only to neurons, which is a detail most descriptions of the brain leave out entirely, mostly because it's less flattering to the story. For most of the 20th century, the working assumption was that glial cells, a name that comes, unglamorously, from the Greek word for glue, existed mainly to hold neurons in place, a kind of biological packing material with no real say in what the brain actually does. That assumption has not aged well. A long-repeated statistic claiming glial cells outnumber neurons 10 to 1 turns out, on careful modern counting, to be roughly wrong. The actual ratio in a human brain is closer to 1 to 1, something on the order of 85 billion glial cells alongside the 86 billion neurons, which is itself strange enough to sit with for a moment. Half of what's in your skull was never a neuron at all, and yet almost everything written about how the brain works talks as if it were. Some of these cells, called oligodendrocytes, wrap themselves around the long arms of neurons in tight repeating segments, like tape wound around a wire, forming a fatty insulating layer called myelin. This isn't decoration. A signal travelling down a myelinated fibre doesn't creep along continuously the way it would down bare wire. It leaps instead from one small uninsulated gap in the wrapping to the next, a process with the genuinely evocative name saltatory conduction, from the Latin for leaping, and it can move a signal more than 50 times faster than an unwrapped fibre of the same width. This is why multiple sclerosis, a disease in which the immune system attacks and strips away myelin, produces such a strange and varied range of symptoms. The neurons underneath are often still intact, still willing, but the signal has lost its ability to leap, and arrives, if it arrives at all, late and degraded, like a voice heard through a wall instead of in the room. Other glial cells, astrocytes, do something closer to groundskeeping, mopping up excess neurotransmitter left in the gap after a signal has crossed, regulating blood flow to busy regions so that the neurons doing the most work get the most fuel, and, more recently discovered, actively influencing which synapses strengthen and which weaken, rather than simply supporting the process from a safe distance. And a third population, microglia, function as the brain's resident immune cells, patrolling constantly, and this is the detail that tends to unsettle people the first time they hear it, physically engulfing and clearing away weak or underused synapses, a process called synaptic pruning that runs in massive waves during childhood and adolescence, quietly deciding which connections were worth keeping and which weren't, without asking the person attached to them for an opinion. A remarkable amount of who you eventually became was decided by cells that were never neurons at all, working while you were doing something else entirely, usually sleeping. Getting into this system at all from outside the body turns out to be its own separate problem, one most of the bloodstream never solves. Wrapped around nearly every blood vessel inside the skull is a tightly sealed layer of cells, joined together with a density of connections rarely found anywhere else in the body, forming what's called the blood-brain barrier. Most molecules circulating in your blood right now, many nutrients, most bacteria, the majority of any medication you might take for an unrelated problem elsewhere in the body simply cannot cross it. Only small, specifically shaped molecules get through, either by slipping directly through the fatty membrane of the barrier cells or by hitching a ride on dedicated transport proteins built to recognize one particular shape and nothing else. Glucose gets a transporter of its own because the brain, despite being about 2% of your total body weight, burns through roughly 20% of your entire glucose supply and cannot be left waiting. Alcohol and caffeine, structurally small and fat-soluble, slip through with almost no resistance at all, which is a large part of why their effects arrive so quickly. The barrier is less like a bouncer checking names at a door and more like a lock that only a small number of specifically cut keys were ever made to fit, built to keep the internal chemistry of this system, the exact balance of ions, glucose, and signaling molecules that everything described so far depends on, from being disturbed by whatever happens to be passing through the rest of the body at any given moment. Now take that hum, and everything quietly maintaining it, and ask what happens when one of those internal ripples finally crosses the threshold and actually fires. Because the moment a neuron does decide, in its blunt, thresholded all-or-nothing way, to send a signal, that signal runs almost immediately into a wall it cannot climb over. For a long stretch of the 19th century, the leading scientific opinion was that there was no wall at all, that the nervous system was one continuous tangled net, cell fused directly to cell, current flowing through without interruption, the way electricity flows through a length of copper wire. This was Camillo Golgi's position, and he held it with enough conviction to share a Nobel Prize for research that, in a genuinely strange twist, ended up disproving his own theory. It was the Spanish neuroanatomist Santiago Ramón y Cajal, staring for years through a microscope at tissue stained with Golgi's own technique, who argued the opposite, that neurons were separate, individual cells, each with its own boundary, its own membrane, touching but never actually merging with its neighbours. Cajal was right. The two men shared the 1906 Nobel Prize anyway, reportedly barely speaking to each other at the ceremony, one of them having spent his career proving the other one wrong using the other one's own methods. What Cajal couldn't see, because the microscopes of his era weren't powerful enough, was exactly how large that gap really was, and it wasn't until electron microscopy arrived, decades later, that anyone could actually look at the space between two neurons and measure it. 20 to 40 nanometers. If you scaled that gap up until it was the width of a football field, a human hair, scaled by the same proportion, would still be enormous standing next to it. And yet, at the scale it actually exists, nothing electrical simply strolls across. A very small number of connections in your nervous system cheat this problem entirely. These are called electrical synapses or gap junctions, and they work almost exactly the way the old 19th century picture imagined. Actual physical channels bridging two cells, letting current pass directly and almost instantly. No delay, no chemical intermediary. They're fast and they're useful for a narrow set of jobs where speed matters more than nuance. Certain reflexes, certain rhythm-generating circuits, places where the nervous system needs every cell in a group to fire together immediately with no negotiation. But they're rare. The overwhelming majority of the connections in your brain, well over 90% by most estimates, do it the hard way, chemically, across an open gap, with no direct physical bridge at all. Here is what that actually looks like, close up, at the level nobody ever gets to witness directly. The electrical signal races down the neuron's long arm and arrives at a slightly swollen structure at its very end, packed with small membrane-bound sacs called vesicles, each one loaded with neurotransmitter. The arrival of that electrical pulse forces open a set of channels that let calcium ions flood into the terminal, and it's specifically that calcium, not the electrical signal itself, that triggers what happens next. A set of proteins with a genuinely evocative name, snare proteins, physically zipper the vesicle's membrane to the cell's outer membrane, pulling them together until they fuse, and the vesicle essentially turns itself inside out, dumping its chemical contents into the gap in a fraction of a millisecond. This entire process, electrical signal to calcium influx to vesicle fusion to chemical release, happens in under a millisecond, one of the fastest biological events your body performs, and it happens, again, something on the order of 100 trillion times across your brain in any given second you're alive. It shouldn't work, and there's a specific reason it barely does. Neurotransmitter release at any single synapse on any single occasion is not guaranteed. British-German physiologist Bernhard Katz spent much of his career, work that eventually won him a Nobel Prize of his own, discovering that neurotransmitter isn't released in some smooth, continuous, dial-it-up or down way. It's released in discrete packets, quanta, whole vesicles firing or not firing, essentially a coin flip weighted differently at every synapse in your brain. Some synapses release with near certainty every time. Others, especially the ones doing the most delicate high-precision work, might succeed less than half the time an electrical signal arrives, the message simply failing to make the jump. No chemical spray, no signal, nothing. The synaptic equivalent of picking up a phone and getting silence on the other end, and never knowing whether anyone was even trying to call. It's worth putting a number on the sheer scale of what's actually happening underneath all of this, if only because most numbers this large stop meaning anything past a certain point. Current estimates place the total number of synapses in a single adult human brain somewhere around 100 trillion, though some methods push the figure meaningfully higher and others meaningfully lower, depending on exactly how a synapse gets counted and which regions get sampled. Even the more conservative estimates put the number of possible connections well beyond anything a single mind could ever hold as an intuitive quantity, closer, in scale, to counting stars than to counting anything you'd find in an ordinary room. And every one of those 100 trillion connections is, at this exact moment, running the same basic, unreliable, probabilistic chemical process just described. A gap, a chemical crossing, a coin flip weighted differently every time. The sheer number doesn't make any single connection more dependable. It just means the overall system can afford, comfortably, for an enormous number of individual coin flips to come up wrong. Your brain compensates for this constant low-grade unreliability the same way any well-engineered system compensates for an unreliable component, with redundancy. Most meaningful signals in your nervous system aren't carried by a single synapse at all, but by dozens or hundreds of parallel connections firing in overlapping patterns so that a handful of failed handoffs barely register against the larger chorus. But the failures are always there, underneath, a constant quiet fizzle of messages that simply never arrived, absorbed into the noise before you ever noticed them missing. And once a message does get across, once the chemical actually reaches the other side of that impossible little gap, what it delivers is nothing like a word. There is a temptation, talking about any of this, to reach for the word language, and it's not entirely wrong, but it flatters the system more than it deserves. A single molecule of neurotransmitter docking into a single receptor isn't saying anything with content. It's closer to a single vote, yes or no, more or less, now or not now, cast into a tally that only means something once thousands of other votes have been cast alongside it. Dopamine, despite a decade of casual internet shorthand calling it the pleasure chemical, behaves much more precisely as a signal of prediction error, the gap between what you expected and what actually happened, spiking when something turns out better than expected, and dipping, sometimes sharply, when it turns out worse, which is a large part of why the second bite of something delicious never quite matches the first. Your dopamine system has already, silently, revised its prediction downward. Serotonin does something far less dramatic and far more constant, showing up in patience, in mood regulation, in the slow background tone underneath a day, rather than in any of its highlights. Acetylcholine handles a strange dual life, driving muscle contraction at the edges of the nervous system while, up in the cortex, playing a central role in attention and learning, tuning how sharply the brain listens to new information. Norepinephrine sharpens arousal and vigilance, the chemical signature of a system deciding urgently that something now matters more than it did a second ago. GABA mostly tells cells to quiet down, a chemical hand pressed gently on another cell's shoulder saying not now. Glutamate does the opposite, and does it constantly. It's the most abundant excitatory neurotransmitter in your entire nervous system, doing the overwhelming bulk of the actual moment-to-moment work while flashier chemicals get all the attention in conversation. That same prediction error signal carried by dopamine has a darker structural vulnerability built into it, one that a great deal of modern addiction research has been built around understanding. Certain substances and behaviors are capable of triggering a dopamine surge far larger and far more reliable than almost anything the brain would otherwise encounter through ordinary experience. Larger than food, larger than most ordinary social reward, arriving with an intensity and a directness the system was never really calibrated to expect. Repeated exposure to a surge that size doesn't leave the underlying system unchanged. The brain, doing what it does everywhere else in this story, adjusts its baseline expectations to match, often by reducing the number or sensitivity of its own dopamine receptors, a process researchers call down-regulation, which means the same experience that used to produce a strong reward signal now produces a smaller one, and ordinary everyday sources of reward, food, company, small accomplishments, start to feel comparatively flat by contrast, less capable of reaching a system that has quietly recalibrated its own sense of what a normal, dopamine-rich moment is supposed to feel like. It's a genuinely large and still developing area of research, but the underlying shape of it connects directly back to something already covered here. A system built to track the difference between expectation and reality, quietly rewriting its own expectations in response to what it's repeatedly shown, whether or not that's actually good for the person carrying it. There's at least one more chemical worth adding to the earlier list, if only because its reputation has suffered from roughly the same oversimplification dopamine has. Oxytocin, released during physical touch, childbirth, breastfeeding, and close social bonding of almost any kind, picked up a nickname in popular science writing, the love hormone, that flattens a considerably more complicated picture. Careful research has found oxytocin increasing trust and generosity in some experimental contexts, and increasing suspicion, favoritism toward one's own group, and even aggression toward outsiders in others, depending heavily on the social situation it's released into. Rather than a simple warmth switch, it behaves more like a volume knob turned up on whatever social significance a given moment already carries, amplifying bonding in a bonding context and amplifying defensiveness in a threatening one, doing considerably less on its own than the tidy nickname suggests. None of these chemicals alone means anything. Meaning only appears in the crowd, in the combination, in which specific chemicals arrive at which specific receptors, on which specific cells, layered over thousands of simultaneous local decisions, happening far too fast and far too distributed for any single molecule to carry the weight of what eventually surfaces as a thought. And even after a chemical does its job, delivering its message, most of it doesn't simply sit there in the gap. It gets pulled back, reabsorbed into the neuron that released it in a process called reuptake, cleared away within milliseconds, the conversation resetting itself almost as fast as it happened, ready to say something new a moment later. Not all of this conversation happens where you'd expect it to either. Somewhere below your ribs, wrapped around the length of your digestive tract, there is a mesh of more than 500 million neurons, more than exist in the entire spinal cord, forming what physiologists sometimes call, only half-jokingly, a second brain. It can't compose a thought, and it doesn't generate anything resembling language, but it runs an enormous amount of independent processing entirely on its own, coordinating the slow muscular waves that move food through you without waiting for instructions from the skull at all. It communicates with the brain above it mostly through a single long structure called the vagus nerve, and the traffic on that line is lopsided in a way that surprises most people the first time they hear it. Roughly 80% of the signals travelling along the vagus nerve run upward, from gut to brain, not the other direction. Your stomach is not simply receiving orders, it is reporting constantly, and a growing body of research suggests that some portion of what gets filed upstream as mood, a vague unease, a sense that something is off, the specific flavour of dread that shows up before you've consciously identified a reason for it, may originate down there in a structure most people never think of as having an opinion at all. The phrase gut feeling turns out to be a great deal less metaphorical than it sounds, so the words genuinely have no mouths wherever they're coming from. Whatever your brain is telling itself moment to moment, it isn't telling itself in anything resembling language as you'd recognize it. Not in sentences, not even in anything you could meaningfully call symbols. It's telling itself in bursts and gaps and probabilistic chemical suggestions, in patterns of on and off happening faster than any single conscious thought could ever hope to track directly. And somehow, out of that, out of something structurally much closer to static than to speech, you get sentences, you get memories with a specific emotional temperature attached to them. You get the aching, oddly cinematic clarity of remembering an argument from 4 years ago, nearly word for word at 2 a.m, for no reason you could ever explain to someone else. There's a piece of this story that tends to get skipped, maybe because it's less visually dramatic than gaps and chemical sprays, but it might be doing more of the actual interpretive work than anything mentioned so far. Timing, not just whether a neuron fires, when exactly relative to everything firing around it. Picture two neurons firing at roughly the same average rate, say 50 pulses a second each. Counted crudely, by volume alone, you'd say they were transmitting the same information. But if one of them consistently fires its pulses a few milliseconds ahead of the other, and something downstream is specifically built to notice that near simultaneity, then the actual message isn't hiding in how often either neuron fires at all. It's hiding in the gap between two events that, to a casual observer counting pulses, would look completely identical. Researchers call this temporal coding, and your nervous system turns out to be extraordinarily good at reading it. In some circuits, dramatically better at reading timing than at reading raw firing rate. The clearest, almost theatrical demonstration of this lives inside a barn owl's skull. A barn owl hunting at night, with no useful light to see by, locates a mouse rustling in grass entirely by sound, and it does this with an accuracy that seems to border on unfair, pinpointing a source in complete darkness to within a couple of degrees. It manages this because its brain has a dedicated circuit that compares the arrival time of a sound at its left ear versus its right ear, and the differences it's comparing are measured in millionths of a second, sound reaching one ear a few dozen microseconds before the other, depending on the angle. Human hearing runs a strikingly similar circuit, at a similarly absurd level of precision. The difference in arrival time between your two ears, sometimes as small as 10 millionths of a second, is how your brain locates where a voice is coming from in a room without you ever consciously calculating anything. There is no dictionary translating that specific interval into that specific direction. There's only a circuit built over evolutionary time to treat timing itself as meaning. This shows up again at a completely different scale in the rhythms your entire brain generates when large populations of neurons fire together in coordinated waves. Brain waves, though the word undersells how functionally important these rhythms actually are. Slower rhythms in the theta range show up prominently during memory formation and spatial navigation. Faster rhythms in the gamma range tend to appear when the brain is binding together separate pieces of sensory information. The color of an object, its shape, its motion, all processed in genuinely separate regions, into one single, unified perceptual experience. This is sometimes called the binding problem, and one of the leading explanations for how it gets solved has nothing to do with location at all. It's synchrony. Regions on opposite sides of the brain, representing completely different features of the same object, seem to fire in step with each other, in time, and that shared rhythm may be exactly what tells the rest of the system these signals belong together, even though nothing about their physical location says so. There is no grammar here in the way a sentence has grammar. No fixed rules mapping a specific rhythm onto a specific meaning the way a word maps onto a definition. There's only pattern, arriving and arriving and arriving, a structure built entirely out of when, rather than what, assembling something that eventually reaches you already dressed up as a complete, unified thought, with every trace of the timing that built it quietly stripped away before it arrives. Timing runs at a slower register too, one your body keeps without any organ you could point to and call a clock. Deep in their hypothalamus, a cluster of roughly 20,000 neurons called the suprachiasmatic nucleus, a name that sounds like an apology for how specific it needs to be, functions as something close to a master timekeeper, running on a genetically encoded loop that, left completely alone, without sunlight or clocks or any outside cue at all, still cycles at close to 24 hours on its own. People who have volunteered to live for weeks in deep caves or sealed underground bunkers, with no access to daylight, no clocks, nothing to tell them what hour it is, still tend to fall asleep and wake on a rhythm close to a day's length, drifting gradually later or earlier without ever losing the underlying pulse entirely. That rhythm gets tuned day to day by a small population of light-sensing cells in the retina that were only properly identified in the early 2000s, cells that don't contribute meaningfully to what you'd call seeing at all, but instead contain a light-sensitive pigment called melanopsin and report, directly and constantly, on ambient brightness to that clock deep in the hypothalamus, which uses the information to adjust the release of melatonin, nudging your entire system to feel like night is approaching or morning has arrived. You have never once consciously checked what time it is using this system. It checks for you, constantly, using a sense you don't even have a name for in ordinary conversation, running underneath everything else you'd call perception. That same 24-hour loop doesn't run at quite the same pace in everyone, and the differences turn out to have a real genetic basis rather than being simply a matter of habit or discipline. Researchers have identified mutations in genes with names like PER2 and PER3, short for period, part of the same family of clock genes that keeps the suprachiasmatic nucleus running on its own, even in total isolation, that measurably shift a person's natural sleep-wake timing earlier or later, producing what researchers call chronotype, the broad, largely inherited tendency toward being a morning person or a night person. People with certain variants of these genes fall asleep and wake naturally hours earlier than average, a condition called familial advanced sleep phase, while others run measurably later, and neither pattern is simply a matter of willpower straining against a fixed, identical internal clock. Two people can be running the same basic circadian machinery, tuned by the same light-sensing retinal cells, and still land on meaningfully different local times, for reasons written into their genes well before either of them ever set an alarm. And here is where the story stops behaving the way you might expect it to, because everything up to this point has had at least the shape of a chain you could follow. Signal, gap, chemical, timing, one step logically leading to the next. And if you followed that chain far enough, it would be reasonable to expect it to arrive somewhere. A destination. A desk, metaphorically speaking, where all these messages finally land, get read, and get turned into a decision, a thought, an experience. There isn't one. And this isn't a gap in current science waiting to be filled by a better microscope. It's closer to the actual, current, best supported understanding of how a brain produces something like a single, unified experience out of an almost incomprehensibly large number of small, local, chemical conversations happening without any central authority overseeing any of it. No single neuron knows what you're thinking right now. No single brain region contains you, full stop, in the way a folder contains a document. Even regions that neuroscience has mapped with real confidence, a patch of frontal cortex heavily involved in planning, a patch of temporal cortex heavily involved in language, do their jobs only in constant overlapping conversation with regions on the opposite side of your skull, relayed through white matter tracts that, if you unspooled and measured every connection in a single human brain, would stretch, by some estimates, well over 100,000 miles. 100,000 miles of internal wiring, and no headquarters anywhere along any of it. The specific way your own nervous system refuses to have a single headquarters starts to look considerably less strange once you look at how differently the same basic problem gets solved elsewhere in the animal kingdom. An octopus has roughly 500 million neurons, in the same general range as a dog, but only about a third of them sit inside its actual brain. The rest, the majority, are distributed directly into its 8 arms, each one containing enough independent neural processing to continue reaching, grasping, and even reacting to a pinch for a period of time after being surgically separated from the rest of the animal, a genuinely unsettling finding from decades of comparative neuroscience research. An octopus arm can, in a real functional sense, taste what it's touching, and begin responding to it before any signal has travelled back to what would ordinarily be called the brain at all. Some researchers studying octopus cognition have started describing the animal less as a single mind operating 8 limbs, and more as something closer to a loosely federated collection of semi-independent processing centres, coordinated but not strictly commanded by the central mass behind its eyes. It's an extreme, almost cartoonish version of exactly the same underlying principle already sitting in your own skull. Distributed processing, no single office, meaning assembled locally and pieced together afterward. Just built by evolution into a shape strange enough that nobody could ever mistake it for having a tidy chain of command. Language itself, the very thing being used to describe all of this, offers some of the oldest and most direct evidence that there was never a single control room to begin with. In 1861, French physician Paul Broca examined a patient, known for decades afterward mostly by a nickname derived from the only syllable he could reliably still say, who had lost the ability to produce fluent speech following a stroke, despite being able to understand language perfectly well, and despite having no apparent damage to his mouth, tongue, or vocal cords. An autopsy after the patient's death revealed damage concentrated in a specific patch of the frontal lobe, now known as Broca's area, and the finding helped establish, for the first time with real anatomical evidence, that specific mental functions could be tied to specific physical locations in the brain, rather than being distributed uniformly, formless, throughout the whole organ. A decade later, German neurologist Karl Wernicke described the near-mirror image of this condition. Patients with damage to a different region, further back in the temporal lobe, who spoke fluently, in grammatically smooth sentences, with normal rhythm and intonation, and said almost nothing coherent, stringing together real words in combinations that carried no retrievable meaning, while remaining largely unable to understand language spoken to them either. Producing speech and understanding it, it turned out, were not one function housed in one place. They were 2 separate systems, connected by a bundle of fibers between them, each capable of failing completely while leaving the other one intact. Damage the connecting bundle itself rather than either endpoint, and a rarer condition called conduction aphasia appears. A person who can speak fluently and understand what's said to them perfectly well, yet cannot repeat a sentence someone just said back to them. The 2 working systems suddenly unable to pass information directly between each other, like 2 fluent speakers standing in adjoining rooms with the door between them jammed shut. Language's lopsided geography lines up loosely, with an even more visible kind of lateralization. Something close to 90% of people are right-handed, a ratio that has held remarkably steady across cultures and centuries, and that correlates, though imperfectly, with left hemisphere language dominance. Most right-handed people process language predominantly in the left hemisphere, while left-handed people show a more varied, less predictable pattern, sometimes left-dominant, sometimes right-dominant, sometimes meaningfully split between the two. Why handedness itself skews so consistently in one direction across the entire species remains only partially understood, tangled up with genetics, prenatal hormone exposure, and a fair amount of ongoing scientific disagreement about how much of any of it is actually inherited in a straightforward way. There is unsettling direct evidence for exactly how distributed this really is, and some of the clearest evidence came from an unlikely source, a set of patients in the 1960s and 70s who, to control severe epileptic seizures, had the thick bundle of fibers connecting their brain's two hemispheres surgically cut. Roger Sperry and Michael Gazzaniga studied these patients for decades, and what they found was strange enough to reshape how neuroscience talks about the self. Show one of these patients an image in their left visual field only, information that reaches solely the right hemisphere, and the patient speaking half of the brain, usually the left, has no idea what was shown, and will confidently, sincerely report seeing nothing. Ask the patient's left hand, controlled by the silent right hemisphere, to point at something related to the image it just saw, and it will point correctly, while the mouth, driven by the other half of the same skull, insists it doesn't know why the hand just did that. Two systems, one skull, each generating what looks, from close enough, like its own thread of awareness, and no evidence anywhere in these experiments of a single overseeing self sitting above both of them, watching, deciding, reconciling the two. Gazzaniga went on to describe something he called the interpreter, a tendency of the left hemisphere in particular to generate confident, fluent explanations for behavior it didn't actually cause, filling in gaps with a story rather than admitting it doesn't know. This might be one of the most quietly important facts anyone has ever pulled out of a neuroscience lab. Your brain would rather invent a reasonable-sounding explanation than sit with a gap, which raises an uncomfortable, useful question about every confident explanation you've ever given yourself for why you did something. Some unknown portion of those explanations may not be reports at all. They may be inventions generated after the fact by a system built specifically to never say, "I don't know." It's worth pausing here to correct something. The split brain research is often inaccurately credited with proving. A popular idea holds that the left hemisphere is the logical, analytical half, and the right hemisphere the creative, intuitive half, and that people themselves can be sorted into left-brained or right-brained types based on which one supposedly dominates. This turns out not to hold up under careful testing. A large 2013 imaging study examining over 1,000 brains found no evidence of individuals having a consistently stronger, globally dominant hemisphere overall. Both sides showed roughly balanced network activity across the whole group, with no meaningful correlation to personality traits like creativity or logical thinking. What is real, and what the split-brain patients demonstrated clearly, is that specific functions, language production, certain aspects of spatial attention, do lean more heavily on one side than the other in most people. What isn't real is the leap from that narrow, specific lateralization to a tidy story about two competing personality types living in one skull. The actual finding is stranger and less flattering than the myth. Not two rival specialists, but one deeply interconnected system that happens to divide certain narrow jobs unevenly, for reasons that have very little to do with whether someone is good at math or good at painting. An entire research effort has grown up specifically around trying to draw the actual complete map implied by all of this, under the name connectomics, the attempt to chart every neural connection in a given nervous system, cell by cell, wire by wire. The only organism whose complete connectome has ever actually been finished is a tiny roundworm called C. elegans, whose entire nervous system contains exactly 302 neurons, a number small enough that researchers were able to trace and publish every single connection by hand using electron microscope images back in 1986. Even at that almost absurdly modest scale, the resulting map ran to hundreds of pages and took over a decade of painstaking manual tracing to complete. A human brain, by comparison, has roughly 280 million times as many neurons, and a genuinely complete human connectome, mapped at the same cell-by-cell resolution, remains, even with modern automated imaging, a project still measured in decades rather than years. The map this whole account has been describing in pieces, the gaps, the chemicals, the timing, the total absence of a headquarters, has never actually been drawn in full for a single human brain by anyone, ever. Everything covered here is built from fragments of a map nobody has finished. That tendency to fill gaps rather than sit inside them isn't limited to explaining your own behavior after the fact. A growing framework in neuroscience, often called predictive processing, argues that filling gaps might be close to the brain's entire operating strategy, running constantly, not just in the aftermath of an action, but underneath every second of ordinary perception. On this account, your brain doesn't wait passively for sensory information to arrive and then figure out what it means. It runs ahead of the data, generating a constant stream of predictions about what it expects to sense next, and what actually reaches your conscious awareness is not the raw sensory signal itself, but something closer to the mismatch, the difference between what was predicted and what actually showed up, corrected on the fly, faster than you could ever consciously track it happening. Vision offers some of the most persuasive evidence for this. Show someone a photograph of a face lit from below in a way that inverts the usual pattern of light and shadow, and even people who know exactly what they're looking at will often perceive the face as a mask curving the wrong way, hollow instead of convex, a genuinely famous demonstration called the hollow face illusion, because a lifetime of prior expectation about how faces are shaped overrides the actual shadow information arriving at the eye. You're not seeing the face in front of you so much as you are seeing your own best guess about it, gently corrected by whatever evidence made it through. Something related and considerably more contested is the idea of priming, the notion that brief, often unnoticed exposure to a word, image, or concept can measurably shift later behavior or perception in a related direction. Evidence, if true, of predictions being nudged by inputs too faint to ever reach conscious notice. Some forms of priming rest on genuinely solid ground. Exposure to a word measurably speeds up recognition of a closely related word shortly afterward. A well-replicated finding with a clear mechanistic story behind it. Other, flashier claims from the same broad category, that reading words associated with old age makes people walk measurably slower For instance, one of the most widely cited findings in the field for over a decade, have failed to replicate in large, careful follow-up studies, part of a broader reckoning researchers now call the replication crisis, in which a meaningful share of once celebrated psychological findings turned out not to hold up under closer, more rigorous scrutiny. It's a useful, occasionally humbling reminder that not everything published with confidence in this field has actually earned it, and that the honest version of this story includes its share of retracted certainty alongside everything that's held up. Faces get their own dedicated shortcut through this same predictive machinery, which is worth pausing on before moving anywhere else. A small patch of tissue on the underside of the temporal lobe, called the fusiform face area, responds to faces specifically and disproportionately, more strongly than it responds to almost any other category of object, and it does this fast enough that a face gets a preliminary tag stamped on it, roughly 170 milliseconds after it appears, well before you'd say you've consciously recognized who it belongs to. Damage to this region produces a condition called prosopagnosia, face blindness, and it's stranger than the name suggests. People with prosopagnosia aren't seeing blurred or missing features. They can describe a nose, a set of eyes, the exact shape of a mouth, in complete detail. What they can't do is assemble those individually clear pieces into a recognized whole. In some documented cases, people with severe prosopagnosia have failed to recognize their own face in a mirror, correctly identifying every visible feature while remaining entirely unable to say whose face it was. Whatever lets a face collapse instantly into a specific, familiar identity, that flash of it's her before you've consciously catalogued a single feature, is doing something closer to pattern recognition than assembly, and when it breaks, the pieces stay perfectly intact. Only the click is missing. One of the more approachable everyday demonstrations of the brain constantly predicting its own incoming sensations is something almost everyone has already personally confirmed without knowing the mechanism behind it. You cannot effectively tickle yourself. Whenever your brain issues a motor command, reach out, move this finger, touch that spot, it doesn't just send the instruction to your muscles, it also generates an internal copy of that same instruction, called an efference copy, and uses it to predict, in advance, exactly what sensation the resulting movement should produce. When your own finger touches your own ribs, the actual sensory signal arriving matches the prediction almost perfectly, and a well-predicted sensation gets partially dampened before it ever reaches full conscious awareness. Precisely the same filtering strategy already at work in ordinary visual expectation. A ticklish sensation from someone else's hand carries no matching prediction at all, arrives completely unforecast, and lands with its full, unfiltered intensity. Researchers have demonstrated this directly, using simple mechanical devices that let a person trigger a tickling sensation on their own hand with a slight, deliberately inserted delay between their own action and the resulting touch. And the longer that delay, the more ticklish and less predictable the sensation becomes, matching almost exactly the growing mismatch between prediction and actual arrival. Even the sense of being tickled, it turns out, is really a report on how well your brain saw something coming. This same filtering shows up in a far more mundane and, once you notice it, far more unsettling way in how little of what's actually in front of you ever makes it into conscious awareness at all. In a study that has become one of the most widely repeated demonstrations in the field, psychologists Daniel Simons and Christopher Shabry asked people to watch a short video of two teams passing basketballs and to count carefully how many passes the team in white made. Roughly half the people who watched this video, absorbed in counting, completely failed to notice a person in a full gorilla costume walk directly through the middle of the scene, pause, thump its chest, and walk off. 9 full seconds of screen time in plain view, unhidden, and gone entirely for half the audience. This isn't a trick of the eyes. The gorilla was, in every optical sense, seen. Light from it reached the retina and travelled down the optic nerve exactly the way light from everything else in the frame did. It simply never crossed whatever threshold turned sensory information into something you're aware of, because attention, elsewhere occupied, had already decided it wasn't relevant. Researchers call this inattentional blindness, and a milder, more familiar version of the same filtering happens to you constantly at a loud party, where you can somehow track a single conversation across a room full of overlapping voices and general noise until, without you doing anything differently, your own name spoken across the room by someone else cuts straight through that same wall of noise and lands in your awareness instantly. A phenomenon researchers call, with unusual directness for the field, the cocktail party effect. Your brain is listening to everything. It is only telling you about a sliver. This same filtering shows up in an even more literal way, in a phenomenon researchers call change blindness. Show someone two versions of the same photograph, nearly identical except for one significant difference: a person's hat missing, a large building appearing or disappearing from the skyline. And if the two images simply alternate with a brief blank flash inserted between them, most people take a surprisingly long time to spot the difference. Sometimes many seconds, sometimes never at all without help, even staring directly at the part of the image that changed. The blank flash is enough to disrupt the low-level visual system's usual habit of automatically flagging sudden movement, and without that automatic flag, attention has to search the scene deliberately, piece by piece, rather than simply noticing the change the instant it happens. A closely related limit shows up in time rather than space. Present someone with a rapid stream of letters or images, one target to spot among them, and a second target appearing within roughly half a second after the first is very often missed entirely. A gap researchers call the attentional blink, as though the act of registering the first target uses up a brief window of processing capacity that simply isn't available yet for whatever comes immediately after. That sliver, whatever attention decides to keep, doesn't get held for very long, and it holds less than most people assume. For decades, the standard number taught in introductory psychology was 7, give or take 2, based on a 1956 paper by George Miller that became one of the most cited papers in the field's history. More careful, more recent work, particularly by psychologist Nelson Cowan, suggests the real number, once you strip away the ability to cleverly group information into larger meaningful chunks, is closer to 4. 4 genuinely separate items held in mind, available at once, before something has to drop to make room for something new. This entire holding function leans heavily on the prefrontal cortex, the region just behind your forehead, and that region happens to be the slowest part of the entire brain to finish building itself. still undergoing significant structural maturation into a person's mid-20s, long after most of the rest of the brain has settled into its adult form. It's a large part of why a 15-year-old can understand a risk intellectually, describe it back to you accurately, and still act as though it doesn't apply to them in the moment. Not because the knowledge isn't there, but because the specific hardware that weighs a distant consequence against an immediate impulse is, quite literally, still being built. Sometimes the gap between seeing and knowing you're seeing splits open completely, and one of the strangest documented cases of this is called blind sight. Patients with damage to the primary visual cortex, not the eyes, which remain fully functional, but the first major processing stop in the brain that receives their signal, report sincerely that they cannot see anything at all in the affected part of their visual field. Ask them to guess, though, forced to choose despite insisting there's nothing there to see, whether a light appeared on the left or the right, or which direction a bar of light was tilted, and a meaningful number of these patients perform far better than random chance would predict, sometimes almost as well as someone with ordinary vision, while continuing to insist they're simply guessing. Some of the visual information is clearly still arriving somewhere in the brain and shaping behavior. It just never crosses into anything the person would call seeing. Something structurally similar, run in reverse, happens every time someone goes under general anesthesia. For a long time, the working assumption was that anesthesia simply switches the brain off, quiets it down broadly, the way turning a dial to 0 silences a speaker. Modern neuroimaging tells a more specific and slightly stranger story. During anesthesia, many regions of the brain stay measurably active, sometimes close to their normal waking levels, while the long-range coordination between those regions, the constant crosstalk connecting a patch of visual cortex to a patch of memory circuitry to a patch of planning circuitry, breaks down sharply. The lights, in a sense, mostly stay on. The building simply stops being able to talk to itself in the coordinated way a unified experience seems to require. Underneath even that cortex-wide coordination sits a much older, cruder structure whose entire job is simpler and more binary, deciding whether the rest of the system gets to be conscious at all. The reticular activating system, a diffuse network of neurons running through the brainstem, acts as something close to a master power switch, sending broad, non-specific activating signals up toward the thalamus and cortex, keeping the whole system aroused and awake, or, when damaged by injury or stroke, failing to do so, producing the deep, sustained loss of wakefulness seen in coma. It's a considerably older piece of machinery, evolutionarily, than most of what makes a specific thought recognizably yours, and it's a useful reminder that consciousness isn't only a story about complexity and integration further up the chain. Somewhere far below any of that, something considerably simpler still has to decide, first, whether to turn the lights on at all. And in a small number of documented cases, that internal light has turned out to still be on in patients where nobody expected it. Neuroscientist Adrian Owen and his colleagues, in a study published in 2006, asked a patient diagnosed as being in a persistent vegetative state, showing no outward behavioral signs of awareness whatsoever, to imagine playing tennis, and separately to imagine walking through the rooms of her own house while lying in an fMRI scanner. Healthy volunteers given the same two instructions reliably show activation in two distinct, well-mapped brain regions, one for each imagined task. The patient's scan showed the same two regions activating, on cue, in the same pattern, indistinguishable from a healthy volunteer's. She had no way to speak, no way to move on command, no outward sign that would have told anyone in the room she was following the instructions at all. Later work by the same research group used this exact technique. Tennis to signal yes, housewalking to signal no, to let a small number of similarly unresponsive patients answer simple questions about their own lives. A conversation with no voice and no visible movement, carried entirely by which patch of cortex lit up in response to a silent request. Philosophers have their own name for the deepest version of this problem. David Chalmers called it the hard problem of consciousness, the specific difficulty of explaining why any of this physical activity, any of this firing and chemical crossing and rhythmic synchrony, should be accompanied by subjective experience at all, rather than happening in the dark, mechanically, with nobody home. Neuroscience has made real, substantial progress on what researchers sometimes call the easy problems, mapping which regions correlate with which functions, tracing which circuits light up during which tasks, and essentially no consensus progress at all on the hard problem itself. There are serious competing theories trying to close that gap. Global workspace theory, which pictures consciousness as whatever information gets broadcast widely enough across the brain to be available to many systems at once, a kind of internal spotlight. Integrated information theory, developed largely by Giulio Tononi, which proposes, more mathematically, that consciousness corresponds to how much a system's information is unified, how much the whole exceeds the sum of its separately measured parts. Neither has been proven the way gravity has been proven. Both agree, from entirely different directions on the same underlying shape. There is no office. The moment noise becomes experience is not a location. It may not be a moment at all, in any sense that has a clean edge. It's worth pausing briefly on how any of this gets known at all, because the tools involved are more indirect than the confident language of neuroscience sometimes lets on. Functional MRI, the technology behind most of the brain imaging findings mentioned so far, doesn't actually measure electrical activity, or thought, or anything resembling a signal directly. It measures blood. Specifically, it tracks tiny differences in the magnetic properties of oxygenated versus deoxygenated blood, on the assumption that busier neurons demand more oxygen-rich blood delivered to their local patch of tissue a few seconds later. A delayed, blurry, indirect shadow of the actual electrical event rather than a picture of the event itself. The resulting images, the ones that show a patch of cortex glowing in a particular color during a particular task, are themselves the product of enormous amounts of statistical processing, comparing brain states against each other and highlighting differences that cross a chosen threshold of significance. A threshold that, chosen carelessly, has in the past produced spurious-looking activation in a scan of a dead salmon, a real, published cautionary result researchers still cite to keep each other honest about how easily this kind of data can mislead if the statistics aren't handled with real care. None of this makes the findings described here worthless. It means most of what's presented as settled fact about the brain is closer to the best current interpretation of a blurry, indirect, statistically processed shadow, revised constantly as the tools improve, which is, if anything, a reasonably honest reflection of everything else this particular subject has turned out to be. The ground gets soft here, and it stays soft, and there's no real way around that softness, only through it. Most of what happens inside that soft, ungoverned activity disappears completely. And this is worth saying plainly, because it sounds like it should be sad and mostly isn't. The overwhelming majority of the electrical and chemical activity firing through your brain in any given second leaves no trace whatsoever. It does its small local job and vanishes, the way most of any ordinary day is gone by the time you're lying in bed that night trying to remember what actually happened in it. Not lost exactly, never kept in the first place. But some of it stays, and the way it stays is stranger than most people assume, because your brain does not file memories the way a cabinet files paper, tucked away whole and intact, waiting to be pulled back out unchanged years later. What actually happens, as best as current neuroscience can describe it, is closer to erosion than storage. Canadian psychologist Donald Hebb proposed, back in 1949, a simple, almost embarrassingly obvious-sounding idea that turned out to be one of the load-bearing concepts of the entire field. Neurons that fire together over and over tend to strengthen the connection between them. When this happens repeatedly enough, the physical structure of the synapse itself changes. More receptors appear on the receiving side. The connection becomes measurably, chemically easier to activate the next time, a process called long-term potentiation. It's not unlike the way a path appears across a stretch of open grass, not because anyone planned to build one, but because enough people kept choosing the same shortcut until the ground itself gave in and remembered the route on their behalf. Strengthening a connection is only half of what this system does, and the other half gets mentioned far less often, mostly because it doesn't make for as satisfying a story. Just as repeated, well-timed firing between two neurons can strengthen the synapse between them. A mismatch in timing, one cell firing consistently without the other, or firing just slightly too late to matter, can weaken that same connection instead, a process called long-term depression, working as the quiet, necessary counterweight to everything potentiation builds. Without it, every connection you'd ever formed would simply keep strengthening indefinitely, an increasingly dense, undifferentiated tangle with no way to tell a well-used pathway from a barely used one. Forgetting in this light isn't simply potentiation failing to happen. It's an active process in its own right, a synapse being deliberately allowed to weaken because the timing that once justified its strength stopped showing up. The grass, left unwalked, quietly growing back over. Some researchers have gone further still, proposing that this constant cycle of strengthening and weakening amounts to something like natural selection happening inside a single skull over the course of a single lifetime. Neuroscientist Gerald Edelman called this theory neural Darwinism, arguing that groups of neurons compete for use the way organisms compete for resources, with better suited, more frequently activated groups being reinforced and less useful ones fading. An ongoing selection process shaping the brain's wiring, based on which patterns of activity actually prove useful in the world the brain finds itself in. It's a genuinely influential idea, though not one universally accepted in its strongest form, and it sits at exactly the kind of ambitious unifying scale that's easy to find compelling and hard to fully verify. What isn't in question is the underlying mechanism it's built on top of, the constant, ongoing negotiation between strengthening and weakening already covered here, running right now somewhere behind your eyes, deciding in its own slow way which version of you gets to keep existing tomorrow. There's a study that gets cited constantly in this territory because it's genuinely almost too good, too literal to be true, and yet it is. Researchers scanning the brains of licensed London taxi drivers, people who had spent years memorizing the city's notoriously tangled, unplanned street layout well enough to pass an exam called, without irony, the knowledge, found measurably larger hippocampi, the brain region most closely tied to spatial memory, than in matched non-drivers, and the longer someone had been driving, the larger the difference tended to be. The physical shape of a piece of their brain had shifted slowly, in response to years of repeatedly walking the same mental route, exactly the way a path gets worn into grass by nothing more than repetition. A related but distinct kind of physical reshaping shows up in musicians, and it maps with unusual precision onto exactly which body part gets used the most. In a well-known 1995 study, neuroscientist Thomas Elbert and colleagues used brain imaging to compare the cortical representation of the fingers on string players' left hands, the hand doing the constant, precise fretting work on the neck of a violin or cello, against the same region in non-musicians. The area of cortex devoted to those left-hand fingers was measurably larger in the musicians, and the effect was strongest in those who had started playing earliest in childhood. Exactly the pattern you'd expect if the physical map inside the skull genuinely tracks how much a given patch of body gets used, rather than being fixed at birth and left alone. The same players showed no comparable enlargement on the side representing their right hand, the one doing the broader, less individually differentiated work of bowing. The map isn't just capable of being redrawn, it's constantly, quietly being redrawn, in proportion to whatever you happen to keep asking it to do. This kind of physical reshaping is not limited to people who deliberately practice something for years, and it is not limited either to childhood, though childhood is where it happens fastest and most freely. Certain windows in early development, called critical periods, are marked by an almost startling openness to being shaped by experience. The visual cortex of a young child, for instance, needs ordinary visual input during a specific early window to wire itself properly at all, which is why congenital cataracts, if left uncorrected past a certain age, can produce lasting visual impairment even after the physical cataract itself is removed. The window for wiring had already mostly closed. Some of the numbers involved in this early openness are worth sitting with directly. A newborn's cortex adds new synapses at a blistering pace, and by roughly 2 to 3 years of age, a young child's brain contains noticeably more synaptic connections than an adult's ever will. By some estimates, close to double the density that will remain once adolescence finishes its work. The system doesn't grow more connected as it matures. It grows more connected first, extravagantly, almost wastefully, and then spends the following 2 decades methodically cutting back, keeping whatever got used, and letting the rest quietly disappear, the microglia mentioned earlier doing much of that literal physical clearing away. An adult brain isn't a child's brain with more added to it. It's closer to a child's brain with most of the excess removed. But the brain does not simply lock its doors once that window shuts, and some of the clearest evidence for that comes from people who lost a sense entirely, rather than merely underused it. In individuals blind from an early age, brain imaging consistently shows the visual cortex, the region ordinarily dedicated to processing sight, becoming active during tasks that have nothing to do with seeing at all, reading Braille, processing complex sounds, even certain kinds of language comprehension, as if a large, well-built room sat empty for years, and the rest of the house eventually decided it was foolish to leave it that way, and started using it for something else. A comparable, if usually less dramatic, kind of reorganization shows up after certain strokes, where a region of cortex is damaged and, over months of rehabilitation, neighbouring or even distant regions gradually take up some portion of the lost function, not by regenerating the dead tissue, but by rerouting around it, the way water finds a new path around a fallen branch rather than waiting for the branch to move. Whether entirely new neurons can join this process in an adult brain, rather than only rewiring the connections between existing ones, remains one of the more genuinely unsettled questions in the field. For decades, the standard teaching was an absolute no. You were born with essentially all the neurons you would ever have, and the number could only decline from there. That absolute has softened considerably since. Evidence for ongoing neurogenesis, new neuron growth in the adult hippocampus, specifically has been reported by multiple research groups, though other careful, well-conducted studies using different methods have failed to find convincing evidence of it in older adult brains, and the disagreement, as of the most recent research, has not been fully resolved. It is one of the rare places in this entire territory where the honest answer is still we don't fully know yet, offered without embarrassment, because that's what the evidence currently supports. In 1953, a 27-year-old man named Henry Molaison, referred to for decades in the scientific literature only as Patient HM, his full identity kept private until after his death, underwent an experimental surgery to control severe, debilitating epileptic seizures, in which surgeons removed large portions of both his hippocampi, the structures already discussed here as central to memory formation. The seizures improved. What happened afterward reshaped the entire field's understanding of what memory actually is. HM could still hold a conversation, still recall most of his childhood and the years leading up to the surgery in reasonable detail, still speak and reason and recognize old friends. But from that day forward, he was almost entirely unable to form new explicit memories. He could meet the same researcher, Brenda Milner, day after day for years, and greet her each time as a complete stranger, with no memory of ever having spoken to her before, despite decades of regular visits. What made HM's case genuinely transformative, rather than simply tragic, was a second, quieter finding buried inside the first. Researchers asked him to practice a task called mirror tracing, drawing the outline of a star while watching his hand's reflection in a mirror, a skill that requires the brain to essentially reverse its usual coordination between what the eyes see and what the hand does. Session after session, HM's performance on this task improved steadily, exactly the way anyone's would with practice, the lines growing straighter, the errors growing fewer. And each time, before beginning a new session, he reported no memory whatsoever of ever having done the task before, sincerely surprised to be handed the star and the mirror again. His hands were learning. The rest of him had no idea. This became one of the clearest pieces of evidence that memory was never a single system to begin with, that the explicit, tellable kind of memory, the kind that lets you say, "I remember doing this," and the implicit procedural kind, the kind that simply lives in a skill without ever needing to be consciously recalled, run through largely separate machinery in the brain, capable of being cleanly split apart by a single surgery, one intact and one gone in the same person at the same time. None of what does get kept is stored the way a photograph is stored, intact, waiting. It's stored the way a habit is stored, easier to walk than to explain, prone to fading if left unused long enough, and this is the part that unsettles people the most once they learn it, reconstructed rather than replayed every single time it's recalled. Retrieving a memory doesn't pull a fixed file out of storage unchanged. It rebuilds the memory on the spot out of whatever fragments are still intact, and in the process, subtly, sometimes not so subtly, rewrites it slightly before filing it away again. This is a large part of why eyewitness memory is so notoriously unreliable, and why the memory you have of an event from 10 years ago may share only a loose family resemblance with the actual event, even though it feels, from the inside, exactly as solid and exactly as certain as it did the first time. And a good deal of this reconstruction work happens specifically while you're unconscious. Sleep is not the brain simply idling. During certain stages, particularly slow-wave sleep, patterns of activity recorded during the day appear to replay, compressed, at many times their original speed in the hippocampus and cortex, a kind of nightly rehearsal that seems to be central to which memories get consolidated into longer-term storage and which quietly fade. During a different stage entirely, REM sleep, the stage most reliably associated with vivid dreaming, large portions of the brain show levels of activity nearly indistinguishable from being fully awake, while the body itself is chemically paralyzed, unable to act out whatever the mind has constructed for it. That same chemical paralysis occasionally slips out of sync with the rest of the waking process, and when it does, the result is one of the more genuinely unsettling experiences a nervous system can produce. Sleep paralysis occurs when a person's consciousness surfaces into full waking awareness, eyes sometimes even open, fully able to perceive the actual room around them, while the muscular paralysis that belongs to REM sleep hasn't yet switched off. The person is, in every meaningful sense, awake. They simply cannot move, sometimes cannot speak, sometimes cannot draw a full breath, and this combination, for reasons still not entirely settled, is frequently accompanied by vivid, often frightening hallucinations layered directly onto the real, correctly perceived room, a sensed presence standing nearby, a weight pressing on the chest, shapes at the edge of vision that aren't there and yet feel entirely convincingly present. It typically resolves within a minute or two on its own, the paralysis catching up to the rest of the system. A brief moment where two states that are supposed to be strictly sequential, fully asleep or fully awake, get stuck briefly overlapping instead. Some of that same nightly replay work seems to specifically favour a looser, more associative kind of thinking than waking logic usually allows. Researchers studying problem-solving before and after a night's sleep have found that people are considerably more likely to spot a hidden, non-obvious solution to a puzzle after sleeping on it than after an equivalent stretch of wakefulness, even when neither group is told the puzzle contains a hidden shortcut at all. REM Sleep in particular appears to loosen the usual rules about which memories and concepts are allowed to connect to which others, recombining fragments from recent experience into pairings a fully awake, rule-following mind would ordinarily keep separate. The old advice to sleep on a difficult problem turns out to have a real, if only partially understood mechanism behind it. Not rest exactly, but a specific kind of unsupervised overnight recombination that waking thought, tethered more tightly to logic and relevance, doesn't seem to perform nearly as freely. Researchers have proposed a range of explanations for why dreaming happens at all, from an older idea that dreams are simply the cortex trying to make loose narrative sense out of essentially random activity firing up from the brainstem, to newer proposals suggesting dreams function as a kind of low-stakes rehearsal space for processing threat and emotion, running simulations of difficult scenarios in an environment where nothing that happens can actually hurt you. None of these explanations has won outright, and it remains, honestly, one of the stranger unresolved territories in the entire field. A brain fully lit up, generating something that feels, from the inside, exactly as real and as consequential as waking life, while producing nothing that will ever leave the skull it happened in. You are, in a very real sense, still doing homework while you sleep, on material you never consciously chose to study, some of it dressed up, for reasons still not fully understood, as an entirely different life. One sense in particular gets a more direct line into this whole reconstructive machinery than any of the others, and it's the one most people think about the least. Every other sense, sight, sound, touch, taste, first passes through a structure called the thalamus, a kind of central relay station that filters and redirects incoming information before sending it onto the cortex for further processing. Smell skips this step almost entirely. Signals from the receptors lining the inside of your nose travel more or less directly to the olfactory bulb, and from there straight into the limbic system, the same tightly interconnected set of structures that houses the amygdala and large parts of the memory system, with barely any of the filtering layer the other senses are forced through. This is very likely why a specific smell, a particular hand soap, a particular kind of rain on hot pavement, the inside of a car nobody has owned in 15 years, can deliver a memory whole and complete and emotionally loaded in a way no photograph or sound ever quite manages, dropping you back into a specific afternoon before you've had any time to brace for it. The route is shorter. There's less machinery standing between the raw signal and the parts of the brain that decide something matters. Sight runs a version of this same chemical relay much earlier, converting light into signal through a process that has nothing intuitive about it once you look closely. The back of your eye is lined with roughly 120 million rod cells, tuned for dim light and motion, and about 6 million cone cells, tuned for color and fine detail, and each one contains a light-sensitive pigment that, when struck by even a single photon, changes shape in a way that triggers a cascading chemical reaction inside the cell, an amplification process so effective that a fully dark-adapted rod cell can, under laboratory conditions, register the arrival of just one single particle of light. That shape change closes ion channels in the cell membrane, which changes the cell's electrical charge, which alters how much neurotransmitter it releases onto the next cell in line. The same basic chemical relay logic already covered here, just running one extra chemical step earlier than an ordinary synapse, converting a particle of light into the same kind of chemical vote everything else in this system understands. There is no moment where light simply becomes an image. It becomes a shape change, then a chemical shift, then an electrical signal, then, eventually after several more relay stations, something that finally resembles seeing, and even then, only the small central patch of your visual field, corresponding to the tiny area of densely packed cone cells at the very back of the eye, is ever in genuinely sharp focus at all. Nearly everything you believe you're seeing clearly, off to the sides of wherever you happen to be looking right now, is being filled in by inference rather than actually resolved in detail. Not everything makes it across, though. Not every message gets through clean, and this part rarely gets mentioned. Maybe because admitting your own hardware has flaws is an uncomfortable thing to say out loud. But you've already met the reason why. Neurotransmitter release, remember, is probabilistic even under ordinary conditions, and that baseline unreliability doesn't stay constant. It shifts. A neuron that has just fired repeatedly in rapid succession temporarily runs low on ready-to-release vesicles and needs a brief window to restock before it can respond at full strength again. A kind of local, momentary fatigue that has nothing to do with willpower and everything to do with basic supply. Stress hormones, released during moments of genuine threat or sustained anxiety, can actively interfere with the hippocampus's ability to properly encode new memories, which is part of why intensely stressful experiences are so often remembered in fragments, vivid in some details and completely absent in others, rather than as a clean, continuous narrative. Sleep deprivation does something similar across the whole system, degrading synaptic reliability broadly enough that entire categories of everyday failure, the word gone right before you needed it, the face you know completely and cannot, for an excruciating 10 seconds, attach a name to, become noticeably more frequent. This happens constantly, in small, forgettable ways, everywhere in your nervous system, and almost none of it registers as anything at all, because your brain runs so many parallel, overlapping versions of the same message that a handful of dropped connections rarely changes the outcome on their own. It's redundancy doing quiet, unglamorous work in the background. But sometimes it does register. That's the thing you walked into a room to get standing in that room now, with absolutely no memory of what it was. A phenomenon researchers have actually studied directly, and, with a certain dry humour, named the doorway effect, finding that the physical act of crossing a threshold seems to reliably disrupt short-term memory in a way that simply continuing to walk in a straight line does not. Your brain appears to use doorways as a kind of natural chapter break, filing away the context of the room you just left the moment you leave it, whether or not you were finished using that context yet. Some of the most instructive dropped signals in the whole nervous system aren't really dropped at all. They're actively rewritten before they ever reach you, and pain is the clearest example. There's an old intuitive assumption that pain works something like a fire alarm wired directly to a damage sensor. Tissue gets hurt, a signal travels straight to the brain. The brain registers the appropriate amount of hurt, done. What actually happens is considerably stranger, and was first properly described by Ronald Melzack and Patrick Wall in 1965 under the name gate control theory. Pain signals travelling up the spinal cord pass through a kind of checkpoint that can be closed, partially or fully, by other incoming signals, including ordinary touch. This is a large part of why rubbing an injury instinctively actually helps a little. The touch signals genuinely compete for the same gate the pain signals are trying to pass through, and sometimes win. It's also why the same physical injury can produce wildly different amounts of reported pain in different circumstances. A soldier wounded in the chaos of battle has, in documented cases, reported barely noticing a wound that would leave most people in a hospital-bound state under ordinary conditions, because the gate, in that moment, for reasons still not entirely mapped, was mostly closed. Phantom limb pain, revisited from this angle, becomes slightly less mysterious and considerably stranger. A limb that no longer exists can still generate agonizing pain, because the neural map that represented it, still very much intact inside the skull, is still sending signals through a gate that no longer has any real damage to report on, misfiring about a body part that isn't there to be examined, believed by the rest of the system anyway. Expectation itself, it turns out, is not merely a mental attitude sitting on top of a separate, purely physical process. In carefully controlled trials, patients given a completely inert sugar pill, but told it's a powerful painkiller, often report a genuine, measurable reduction in pain, not merely a change in how they describe the pain, but detectable changes in brain activity in regions associated with pain processing, and, in some studies, an actual release of the body's own natural opioid-like chemicals, endorphins, triggered by nothing more than belief and context. Researchers have confirmed this mechanism in a fairly direct way. Give someone naloxone, a drug that specifically blocks opioid receptors, alongside that same inert sugar pill, and the placebo effect largely disappears, which strongly suggests the relief wasn't imaginary in any dismissive sense of the word. It was a real chemical event, triggered by expectation rather than injury, using the exact same biological machinery a real painkiller would use. The gate discussed a moment ago, it seems, can be closed not only by a competing touch signal, but by a conviction, sincerely held, that help is already on its way. The same mechanism runs in reverse, and considerably less pleasantly, under the name nocebo. Patients warned, sincerely and often necessarily, about a medication's possible side effects, sometimes go on to experience those exact side effects at rates well above what the inert placebo arm of the same trial would predict, simply from having been told to expect them. In some of the more striking documented cases, patients given completely inactive dummy pills, but told the pills might cause nausea or headache, reported those specific symptoms at rates matching people taking the actual active drug. Expectation, it turns out, doesn't only have the power to ease something real, it has just as much power to manufacture something that wasn't there before. Pain's habit of misreporting isn't limited to phantom limbs. Sometimes it gets the location wrong, even when the damaged tissue is real and present and fully attached to the body it belongs to. A heart attack is one of the clearest examples. The actual damage occurs deep inside the chest, in cardiac muscle, with comparatively few dedicated pain fibers of its own. But people experiencing one very often report pain radiating down the left arm, or in the jaw, regions with no cardiac tissue whatsoever. This happens because pain signals from internal organs and pain signals from certain areas of skin travel up the spinal cord along overlapping shared pathways, converging on some of the same relay neurons before continuing onto the brain. When a signal arrives along one of these shared pathways, the brain, working from limited information and a lifetime of far more frequent skin-based pain than internal organ pain, makes its best statistical guess about the source, and its best guess is very often wrong in a specific, well-documented and occasionally dangerous direction, the whole reconstructive process making a completely reasonable inference that happens, in this particular case, to be dangerously misleading. The map underneath all of this rewriting is worth a slightly closer look because it was drawn, quite literally by hand, decades before any scanner existed. In the 1930s and 40s, Canadian neurosurgeon Wilder Penfield operated on the exposed awake brains of patients undergoing surgery for severe epilepsy, a genuinely strange fact that becomes less alarming once you know the brain itself contains no pain receptors, so patients could remain conscious and responsive under only local anesthetic to the scalp. Penfield touched a mild electrode to different points along a strip of cortex and at each point simply asked the patient what they felt. A tingle in the thumb, a pull in the lips, a twitch somewhere in the leg. Point by careful point, he built a map of which patch of cortex corresponded to which part of the body, and when that map is drawn as a figure, with each body part sized according to how much cortex it was allotted, rather than its actual physical proportions, the result is a strange lopsided little creature, hugely swollen lips and hands and tongue, attached to a comparatively tiny torso and legs, a figure researchers still refer to, half fondly, as the homunculus. Decades later, neuroscientist V. Ramachandran used a plain box fitted with a mirror to intervene directly on that same kind of map in patients with phantom limb pain. A patient would place their intact hand into the box so its mirrored reflection appeared to sit exactly where the missing hand would have been, then move it, opening and closing a fist that, visually, appeared to belong to the missing side. A striking number of patients reported for the first time since the amputation that the phantom hand, clenched for years, aching, unresponsive to anything, finally seemed to relax. Nothing physical had changed. The map itself had simply been shown a picture it was willing to believe. There is one more layer sitting underneath all of this, and it might be the strangest thing your brain does, or it might be the most ordinary thing about you, depending entirely on which direction you're standing in when you look at it. Because your brain does not only talk about the world outside your skull, it talks about itself. This wasn't part of any obvious design, and there's no clean, tidy evolutionary story where a self-monitoring loop was the intended goal from the start. It's more likely that this emerged the way a great many complicated things emerge in biology, as a side effect of enough other systems getting layered on top of each other until a certain kind of feedback became functionally unavoidable. Somewhere underneath the ongoing conversation your brain is having about the room you're currently in, the sounds you're currently hearing, the temperature of your hands right now, there is a second, quieter conversation running about the first conversation. In the early 2000s, neurologist Marcus Rachel and his colleagues noticed something that seemed, at first, like a measurement error. A set of brain regions that consistently became more active, not less, when research subjects were told to simply rest and do nothing in particular, no task, no instructions, just lie still in the scanner and let their mind wander. Far from going quiet, this network, now widely known as the default mode network, lit up more than it did during many active, effortful tasks. It seems to specialize in exactly the kind of activity most people would call daydreaming, remembering the past, imagining the future, running social simulations of what someone else might be thinking, constructing and revisiting a continuous internal narrative about who you are. Checking in on itself was never the plan. It just became, at some point, structurally unavoidable. Once enough smaller feedback loops were already stacked on top of each other. Meditation researchers have found that experienced practitioners tend to show reduced default mode network activity during meditation, alongside a corresponding reduction in the kind of self-referential mental chatter that most people, if you ask them, describe as the background noise of simply being a person, which suggests, indirectly, that at least some portion of that constant internal narration is not a fixed, unavoidable feature of having a brain, but something closer to a habit, a well-worn groove that can, with enough repetition in the other direction, be worn slightly shallower. That same background network involved in daydreaming and self-narration also seems to play a specific, measurable role in the particular kind of thought most people describe as a sudden flash of insight, the answer to a problem you'd stopped consciously working on, arriving unannounced in the shower or on a walk. Researchers Mark Youngbeeman and John Kunios, studying people solving word association puzzles while hooked up to EEG and fMRI, found that moments of sudden insight were reliably preceded, roughly a third of a second beforehand, by a distinctive burst of gamma frequency activity in a specific fold of the right temporal lobe, a burst that didn't appear before answers arrived through slower, more deliberate step-by-step reasoning. Insight on this evidence isn't simply logic running faster under the hood. It looks like a genuinely different mode of arriving at an answer, one that seems to require a certain amount of unfocused, wandering attention to set up properly, which may be part of why so many people report their best ideas showing up specifically when they've stopped trying to have them. A related, though more speculative idea has grown up around what athletes, musicians and writers often describe as a flow state, total absorption in a demanding task, self-consciousness seeming to disappear entirely, time distorting in the opposite direction from a moment of danger, hours passing in what feels like minutes. Some researchers have proposed a hypothesis called transient hypofrontality, suggesting that flow states involve a temporary reduction in activity in parts of the prefrontal cortex responsible for self-monitoring and conscious deliberation, effectively quieting the same self-referential narration discussed here in order to let a well-practiced skill run more directly, with less interference from the part of the brain that usually insists on checking in. It's an appealing idea, and it fits neatly with everything already covered about the self-monitoring loop being a genuine, measurable, quietable system, rather than a fixed, unavoidable feature of thought. It's also, it should be said plainly, still a working hypothesis rather than a settled finding, based on a smaller and less consistent body of evidence than most of what's been covered here, offered less as established fact and more as one of the more interesting open questions currently being tested. Part of what that quieter, self-directed conversation seems to be built on is a steady stream of reports coming from inside the body itself, not just from the outside world. A region called the insula, tucked deep in a fold on each side of the brain, receives near constant signals about your heart rate, your breathing, your gut, your temperature, a kind of internal sensing researchers call interoception, and appears to play a central role in something most people assume is simple and automatic: knowing what you're feeling. it turns out to be considerably less automatic than it feels from the inside. Psychologist Lisa Feldman Barrett and others have argued, based on a substantial body of research, that emotions aren't pre-packaged reactions your brain simply detects, the way a thermometer detects temperature, but are actively constructed on the spot, combining raw interoceptive signals, a racing heart, a tight chest, an unsettled stomach, with context and past experience to produce a specific, named feeling, which is part of why the exact same racing heart, tight chest bodily state can be interpreted, depending entirely on context, as excitement at a concert or dread before a difficult conversation. Your body sends up the same rough weather report either way. Something further upstream decides what to call it. Some of what decides tends to move faster than thought is willing to admit. Neuroscientist Joseph Ledoux spent much of his career mapping how fear specifically gets processed, and found that a threatening signal, the sudden shape of something coiled on a path ahead of you, can reach the amygdala, a small almond-shaped structure buried deep in each temporal lobe, along two separate routes running at two very different speeds. A fast, crude, low-resolution route runs almost directly from the thalamus to the amygdala, skipping the cortex entirely, cheap and quick and prone to false alarms, capable of triggering a flinch, a spike in heart rate, a full-body flood of adrenaline before you've consciously registered anything resembling a coherent thought. A slower, much more careful route runs the same information through the visual cortex first, checking it properly, and arrives at a verdict. Stick, not snake. A beat or two later, sometimes after your body has already reacted to a threat that was never actually there. You have flinched at things that turned out to be nothing more times than you could count, and every one of those flinches happened because some part of you decided correctly by its own standards that being wrong quickly was safer than being right slowly. That same ancient alarm system still triggers a reflex that has mostly outlived its usefulness. A small muscle attached to the base of every hair follicle, the erector pili, contracts under a surge of the same fight-or-flight chemistry the amygdala helps set in motion, pulling each hair upright and raising the small bump of skin around it. Goosebumps, a reflex clearly inherited from a much hairier ancestor, for whom raised fur meant looking larger and more threatening to a predator, or trapping a thicker layer of insulating air against the cold. It still fires, faithfully every time something frightens or moves you deeply, on a body with nowhere near enough hair left for it to do anything useful at all. That same fast, threat-tuned circuitry has a strange relationship with your sense of how much time is actually passing. People who survive genuinely dangerous events, a car accident, a fall from a height, very commonly report that everything seemed to slow down, that they had time to think several distinct thoughts in what turned out to be a fraction of a second. Neuroscientist David Eagleman tested this directly by having volunteers experience a real, if carefully controlled, free fall, a backward drop of over 100 feet into a safety net, while wearing a device on their wrist flashing numbers slightly too fast for ordinary perception to read. If danger genuinely sped up perceptual processing the way it feels like it does, the falling volunteers should have been able to read numbers during the fall that they couldn't read under calm conditions. They couldn't. Their perceptual resolution during the fall was no better than normal. What was different was memory. The frightening experience was laid down far more densely than an ordinary memory, more detail packed into every second, and it's that unusually dense recording that gets played back afterward and interpreted in retrospect as the event itself having taken longer. Time didn't actually slow down while it was happening. Your memory of it was simply built with far more stitched into it than usual, and a memory that dense feels afterward, like it must have taken longer to happen.